on the toilet. It's not lifting the seat. No, she can't actually.
Welcome to MetPhys in Isolation, a new model for seminar series where we can share state-of-the-art science across the globe in a cost-effective, inclusive, and green manner. We've had a competition running at home to accomplish a new feat while the MetPhys 2020 series is ongoing. Cassidy's challenge that she gave herself was to take a photo in front of herself, the Inception challenge every day. She's nailed it so far. The thing I really enjoy is she's adding a new theme. There was a full moon, so there she is as a wolf. Well done, Cassidy. We're all howling at that one. Ooh! And now's your time to take a new photo. My boys at home have decided that during the isolation, it's time to try a new challenge for themselves. And yes, they've decided to learn how to ride a bike. When your mom plays ice hockey, the first thing you learn to do is skate. They've got that under the toe, and now they've learned to ride bikes. That's both of them yesterday, mastering it for the first time. Good on you, boys. And there's little sister in the background. She's been potty training herself, which has been quite a surprise, but we're getting there. Speakers have taken on a challenge for having the best Zoom background. I was getting ready to declare Isaac Harris the winner after our Twitter poll until Kristen came in yesterday and demonstrated you don't need a Zoom background when your background perfectly matches the topic of your talk, and he comes in with the best home gym I've seen. Well done, Kristen, for nailing that. Looking forward to seeing more home challenges, and as I promised, Martin and I will nail that task that we failed at the beginning of the sessions. We are going to get that lift from Dirty Dancing by the end of the seminars. Joshua had this fantastic question a few talks ago, and made me make a new slide to go through how we do this. These talks are live and also being watched in a recorded fashion. However, we can all contribute to discussion. Even if you listen to this talk 3 weeks from now, all you need to do is go on Twitter, use the hashtag MetPhys2020. If you know the speaker's Twitter handle, include that in your tweet, and you can ask any question or make any comment you like. In fact, you can make multiple. There's no limit, and I think that's really nice. And the speakers are getting at those questions pretty quickly and allowing us all some new insight, and it's also kind of recorded for us. So we learned from Sophie some best approaches for stopping metabolism, or how they approach some of those difficult questions, how do they isolate their organelles, or what the timing is of, and all that kind of information we're getting from these question and answer periods. So I think it's really fruitful. So keep that up. I also like the fact that anybody listening from anywhere can ask the speakers a question. And in fact, I do like that we're getting questions through the night over here. I wake up to new questions for the speakers from people who've been watching the recording. So that's pretty fantastic, and I'm glad we're able to do that. So keep that coming. I know the speakers are really enjoying all the feedback and all the comments, and that's a nice way to thank them. So if you don't have Twitter, it's easy to get. I think you can even, going through an Explorer or Safari browser, just go to twitter.com, and in the search bar, without even having a handle, search MetPhys 2020 and see what pops up. So you don't even need to sign in, I don't think, to be able to follow the proceedings. And if you look up the speakers, you'll be able to see things that are addressed directly to them. I am trying to rapidly retweet those with the hashtag. So if you are tweeting a question, try to keep that in there. But if you don't, I'll catch it, and so that everyone can share. And I think that's really great. So thank you, everyone, for your involvement. I think it's great that we keep this going. And you are welcome to not only share questions and answers with MetPhys 2020, but share photos of your dogs and cats and anything that's going on. I think all those kind of sharing moments are helping the community kind of feel that we're not alone, even though we are in isolation. And I think we'll all take the good from that. So yeah, please share freely and enjoy the space for our community. I've said every talk that we really need to thank the speakers. Today, I think I speak for everyone in the community when I send our heartfelt condolences out to Yontja. We're sending love and comfort your way, and we know that it's difficult times ahead, but we're all here for you and we're thinking of you. So today's speaker is Ali Kohan from Pittsburgh, and tomorrow I'm really excited that my mentor for graduate school, Andre Moret, the editor-in-chief of AJP Endometabolism, is going to be giving us a fantastic talk on gut microbiota. So we're kind of finishing the week with some thoughts about the intestines and the gut. So stay tuned tomorrow as well. You'll see these Facebook and YouTube icons. That means that recordings have been uploaded there for your viewing pleasure. If you go to the White Mega Lab webpage, we have a tab called MetPhys 2020 with all the links, including the link to our Spotify playlist. So please share that broadly. Anyone who wants to listen but wasn't able to catch the live feed can get it. Anyone who wants to watch live can. There's no password, and there's no capacity limit to how many people can watch, so please share. Graduate students, undergrads who might be interested in science, your other colleagues who might not be on Twitter, get the word out. I think it's nice that we have this resource and can kind of share our enthusiasm for metabolism in this way. Like I've said the last few weeks, we've got an extended lineup getting us out to April 24. I've learned some exciting news. You'll see that our speaker photos have shifted one, and right next to Ibru Ebe is a photo of an undercover speaker. This is a surprise speaker who is going to do the intro for Ibru. I think on Tuesday next week, everyone should tune in. I'll give some more information early in the week about who that is, but trust me, it's definitely worth tuning in. It's definitely worth sharing. Tuesday is going to be one for the books. I am very, very, very excited about this, and I think you all will be too when you find out who the speaker is going to be. If you'd like to thank me for this series, the way you can go about that is simply, if you have any available funds that you'd like to make a donation with, consider donating to the Pathway to Stop Diabetes program from the American Diabetes Association. I know that they are supporting a lot of great research in our area. I wouldn't be here without support from this program. And I know Celine Riera, who's coming up next week, is supported by them. We also heard from Sophie Treffelet that her postdoc work is supported by the ADA, and I know a lot of other scientists have had their careers launched by them. So it's definitely a mission that's worth supporting. And if you have the means to do so, that would be an honor to us if you did that. If you don't have the means to do so, don't worry about it. This is up here for your viewing pleasure, and you're free to engage and enjoy as you wish. So I mentioned earlier, but tomorrow, my mentor, Andre Moret, from University of Laval in Quebec City, will be presenting his work on the bacteria in human obesity. This is some really exciting work, and I know it's going to be a great talk. So tune in for that. And then without further ado, I'm going to get over to Ali I'm going to make her the host right now. So I will see you all in the Twitter space and tomorrow. Enjoy your day and enjoy the talk. Making Ali the host. Ali, we can see you. I'm going to get rid of me. Stop share. Stop video. Okay.
All right, hopefully we're all good. I'll trust that Phil will give me a text or a call to let me know if he can't hear me. Okay, got the go ahead, so we're all good. Phil, thanks so much for including me in this series of talks. I'm really excited to participate. I want to take a minute and just share my excitement at watching the world turn to the scientific community at this time of crisis. I'm really proud to be a scientist right now. I think for those of you in the audience who are early career, who are undergrads, grad students, postdocs, I think, you know, it's a auspicious time for you. It's a scary time. I want to let you know that there are a lot of people, a lot of faculty, a lot of people on grant committees, people who are reviewing manuscripts. We are thinking about your career. We're thinking about the way that this quarantine and research stoppage is impacting your future. My heart really goes out to people who are on the job market. I think that there is a huge amount of support to try to lift up and support you, especially, again, people who are in early career and transition periods in their career. And so if I can just take a second to say, hang in there. I can't begin to imagine what a difficult time this is about being concerned about your research productivity, but hang in there. I think that there are a lot of allies out there who want to see you succeed and continue in science, because what we do, both basic and translational scientists, is really, really important. I also want to take a second at the beginning of this talk to say that we as a community should be striving for inclusivity. And Phil, I really appreciate the inclusiveness of this seminar series. We should be doing what we can to lift up those around us who are less privileged than ourselves. You know, whether we have kids at home, parents, partners, different life experiences, we are all trying to do science, and we should all, especially those of us in positions of power, be trying to lift up those around us, and especially paying attention to the scientists who are underrepresented in each of our fields. And so I just want to take a second to say that that's really important at this time and at all times for us to use our privilege that way. So now I'm going to hopefully tell you guys a story that we've been working on for the last several years. This is totally unpublished data, and not for lack of trying. We are still trying to figure out where this fits and figure out some mechanism, as well as some of the physiology. But the story I'm going to tell you guys today is about one of the apolipoproteins, ApoC-III, as well as regulatory T cells in the gut, and specifically the way that these two interact to change lipid metabolism. And the thesis really is that what might be good for the heart in terms of changes in lipid metabolism and lipoprotein metabolism may not be good for the gut. As Phil said, I'm here at the University of Pittsburgh. I moved here in September, and so far it's going really well. Okay, let me figure out how to advance here. You can find me on Twitter at Cohen_lab. So I'll start by telling you a little bit about ApoC-III and its role in disease. ApoC-III is one of the apolipoproteins that is made in the liver and in the intestine. And high levels of ApoC-III cause hyperlipidemia in humans by inhibiting triglyceride clearance and extending the postprandial period. I'm going to talk a lot more about that in a bit. But one of the things that we've always known about ApoC-III is its relationship to plasma triglyceride levels. We know that plasma ApoC-III levels are an independent risk factor for cardiovascular morbidities and mortalities. This is also modeled in mouse models that have changes to ApoC-III. Frank Sachs' group also identified HDL containing ApoC-III as being associated with an increased risk of cardiovascular disease rather than cardioprotection, which really jives with the idea that HDL is not a single lipoprotein subspecies, but that there are protective HDL as well as non-protective HDL. And again, ApoC-III on an HDL particle may be a marker of HDL that will not help with cardiovascular disease. Underscoring this role of ApoC-III in the regulation of plasma triglycerides, as well as in the development of cardiovascular disease, there has been a real focus on pharmacological targets, pharmaceuticals that target ApoC-III. And volanesorsen was one of these early pharmaceuticals. And the idea was to use an antisense technology to knock down ApoC-III in the liver and thereby reduce triglyceride levels. And doing this first in people with familial chylomicronemia syndrome. And again, the idea here is simply that ApoC-III is likely a very valid target for reducing plasma triglycerides, especially in people for whom triglycerides are poorly controlled by diet or concomitantly with statins. We know from large epidemiological studies like the multi-ethnic study of atherosclerosis, MESA, that there is an association of ApoC-III content on HDL with cardiovascular disease risk and kidney function. So another strong piece of evidence linking ApoC-III with human disease. And so my lab is really interested in understanding the way that triglyceride metabolism, plasma triglyceride clearance, and ApoC-III work together to change physiology. And so we start with this basic physiological fact. Triglyceride-rich lipoproteins, shown here, chylomicrons and VLDL, are the body's physiological lipid fuel. Chylomicrons come from the intestine in response to a lipid-rich meal, and VLDL are made in the liver and secreted throughout the day. If you look in a basic metabolism and physiology book, you will see that chylomicrons are secreted in the postprandial or fed state, so approximately 2 to 6 hours after a meal, and that VLDL are high during the fasted state. In reality, humans are almost always in the postprandial state. So if you wake up, if you have kids like I do, or a hungry cat like I also have, you might get woken up at 6:15 in the morning by a gentle nudge to get a snack. You have your coffee with cream, maybe you have a bagel with cream cheese. You're eating fat, and so your intestine starts to produce chylomicrons. You eat again at noon, so that's 6 hours since you've had your morning meal. You're still in the postprandial phase. You're going to start making chylomicrons again. You have your afternoon snack, you eat dinner, then you get your kids and your cats to bed, and now you're having your quarantine ice cream, which we've been enjoying each night. So again, you're spending almost the entire day in the postprandial phase. You're making chylomicrons continuously. And at the same time, you're also producing and secreting VLDL. These particles contain at their core triglyceride as well as cholesterol. They have phospholipids and apolipoproteins that emulsify these particles so they can travel in your blood. So again, they've packaged the hydrophobic piece and surrounded that by the apolipoproteins. And those apolipoproteins really mark and manage these particles for their metabolism and clearance. So things like for a chylomicron, ApoB48, ApoC-III, ApoC-II. For VLDL, you'd get ApoB100, ApoA1. There's a whole constellation of apolipoproteins that tell the peripheral tissues, the non-intestinal, non-hepatic tissues, what to do with these lipid-rich particles. And so these are the physiological lipid fuel. This is the way that your tissues see triglyceride and cholesterol, and predominantly triglyceride here. And I want to contrast this to the idea that our tissues see a lot of free fatty acid. So in this case, I'm using free fatty acids bound to bovine serum albumin. So obviously this is a situation in tissue culture, but for most of us who are doing cell culture studies, this is the way that we add lipid to cells. We do it by complexing free fatty acids like arachidonic acid or linoleic acid to albumin. And then we add this complex, which can now dissolve in the culture media, to our cells. And we ask questions about how much of it traverses the membrane and then what it does. And what I want to point out here is how different functionally this free fatty acid complex is from the physiological lipoprotein context. And so again, that's the chylomicron and the VLDL that have these triglyceride moieties, which not only have to dock and bind to the membrane, but then have to be hydrolyzed to their free fatty acid and monoacylglycerol components. And so we're very interested in what these triglyceride-rich lipoproteins do and how they're regulated, how they're involved in disease. And so we know that ApoC-III inhibits the endocytosis of plasma triglyceride-rich lipoproteins. This was really the very first thing that ApoC-III was was determined to have done, which is to inhibit lipoprotein lipase, shown here, LPL, on the capillary endothelium, which acts to hydrolyze triglyceride into free fatty acids that can then be taken up by peripheral tissues. And so plasma C3 was shown through a variety of careful in vitro studies to inhibit LPL. And then more recently, ApoC-III was shown to play a very large role on the low density lipoprotein receptor LDLr in the liver. And plasma ApoC-III inhibits LDLr and inhibits the uptake of these triglyceride-rich lipoproteins. And that was shown by Phil Gortz and crew a few years ago. And what we know now is that the inhibition of LPL probably is about 10% of what ApoC-III does to change plasma lipid levels, whereas the rest is really accomplished by LDLr. So this is the main part of the pathway. We also know that intracellular ApoC-III in hepatocytes increases the secretion of VLDL. And it does this by increasing the rate and the amount of triglyceride that gets loaded onto the nascent VLDL particle in the liver. And so if you combine these two actions, the inhibition of clearance through lipoprotein lipase, the inhibition of clearance through LDLr in the liver, and then the increase in VLDL triglyceride secretion, we see that ApoC-III together will act to raise plasma lipid levels. You will retain remnant triglycerides in blood while also secreting more VLDL. And part of this is how ApoC-III drives plasma lipid levels. As I mentioned in the intro, ApoC-III is also expressed in the intestine. It's expressed mostly in the small intestine, and its level of expression really is highest in the proximal intestine, the duodenum, lowest in the ileum, and the level of expression in intestine is approximately 20% the level of the expression in liver. We started these studies by asking whether ApoC-III would impact chylomicron secretion similarly to the way that it impacts VLDL secretion. And we found through a variety of models and papers that, very surprisingly, ApoC-III does not increase chylomicron secretion. It doesn't increase the rate of secretion or the amount of triglyceride. In fact, ApoC-III appears to delay dietary lipid absorption. And so some really elegant work by my most recent PhD student, Diana Lee, who's now at Duke, she really determined that the role of ApoC-III is in the gut is due to local changes to fatty acid metabolism driven by high levels of ApoC-III at the basolateral membrane. And so I'd encourage you to check her out on Twitter and also check out this paper detailing that mechanism. What we were surprised about was that the role of ApoC-III in the gut so drastically diverged from the role of ApoC-III everywhere else. And as a lab that studies the intestine, I suppose we shouldn't have been so surprised that the body has intestine-specific mechanisms, but it appeared right away that high levels of ApoC-III in the gut, by delaying chylomicron secretion and changing enterocyte fatty acid metabolism, really might be an interesting target and also might tell us a lot about how the gut regulates whole body metabolism. Now, one of the things that we started looking into when we realized that ApoC-III plays such a specific role in the gut was the idea that the ApoC-III gene is associated with inflammatory bowel disease in both mice and humans with no known mechanism. And this really caught our radar because of the specificity of ApoC-III in the gut and inflammatory bowel disease in the gut. And what we know is that a reduction in the ApoC-III gene is one of the largest changes in ileal gene expression between healthy patients and Crohn's disease patients. And in this case, these are pediatric patients. So again, through a variety of RNA-seq, ApoC-III always pops up as one of the largest changes. We also know that the ApoC-III gene and the gene cluster on which it resides, the ApoA1, A4C3 gene cluster, both the expression of the whole cluster and the gene are reduced in the colon and the ileum of adult Crohn's disease patients. And if you look at protein expression in blood during a flare-up, those levels in plasma are reduced. We also know that intestinal epithelial cell specific HNF4 alpha knockout, which is one of the many elements that regulate the A1, A4C3 gene cluster, the knockout of HNF4 alpha can cause spontaneous intestinal inflammation in mice. So, you know, all of this is really correlative. There's certainly no slam dunk here, but we became really interested in understanding whether C3 plays a role in inflammatory bowel disease, and if so, what its role might be. And so this brought us to a pretty simple question. And the question was whether immune cells could endocytose triglyceride-rich lipoproteins. And if they did, what the outcome would be. And so I want to point out a couple key features that we are really interested in on this diagram. One, in green, I'm showing you just a completely generic immune cell. I'm going to talk specifically about T cells and Tregs shortly, but in general, you know, you can consider this a generic cell. Here in red, you can see my depiction of a glucose transporter with glucose uptake. There's the uptake of free fatty acid from a variety of cell culture models like palmitate dissolved in DMSO, or free fatty acid bound to bovine serum albumin. And again, these would either cross the membrane depending on their diluent. DMSO likely just allows the fatty acid to travel through, or free fatty acid bound to albumin might bind something like a CD36 or a fatty acid binding protein type transporter. And so when we think about the landscape of lipid uptake by immune cells, I think many of us are really thinking about this arm of the system. We are really interested in this arm, which had not been shown to be involved in immune cell activation and function. And again, the idea here is functionally, physiologically, almost all of the lipid in your blood is always in the form of these triglyceride-rich lipoproteins. We did a back of the envelope calculation using the American Heart Association target ranges for plasma triglycerides. And what we see is that there's approximately 10 times the amount of acyl substrate, so each of these acyl groups on the triglyceride, present in blood on these lipoproteins compared to the amount of free fatty acid that's bound to albumin, even in the fasting state. So again, that's a back of the envelope calculation, but it tells you a little bit about the size of a difference between the physiological substrate and this, what we would consider a less physiological substrate. And so we asked, do lipoproteins deliver lipid? Do they do it through a receptor? And is this important to immune cells? And does ApoC-III inhibit this, like ApoC-III does in every other tissue that has this mechanism? And so now I'm going to show you some data finally. I want to tell you a little bit about our transgenic mouse model. This is the model that was originally generated by Jan Breslow. It's a human ApoC-III transgenic mouse, so it expresses the human ApoC-III transgene driven by its own promoter. These mice were originally on a mixed background. We've back-crossed them over 8 generations, so they're on a C57 black 6J background. We swap cage bedding in our lab for 2 weeks prior to all experiments. So I want to talk about this for just 1 second. So for immunological studies, the background and the intestinal microbiota are so clearly linked to immune function. And while we cannot make sure that all the microbiota are identical, what we try to do to ameliorate that difference is swapping dirty cage bedding. And so what we do is we take, for 2 weeks, we take the dirty cage bedding from our experimental animals and we put it into the cages with our control animals and vice versa. And that occurs 2 weeks prior to all of our experiments. And that's one way that we try to increase rigor and reproducibility in terms of our experimental animals and our controls. It's not perfect, but it's one approach. I also want to point out that we use male and female litter mates. For all the studies I'm showing you here, we've used mixed cohorts that are aged matched. We have looked, we don't see a big difference between female and males. Obviously, with what's going on with COVID-19 showing that there are such significant differences between the male and female immune responses to virus. I think all of us who are doing immunology work probably have to double down to consider these differences. So we use male and female so that we don't lose those potential changes. So let me tell you a little more about this model. So here is ApoC-III expression on this axis. This is mRNA expression over a variety of tissues. So we'll start here in the liver. In the hatched bar is the endogenous mouse ApoC-III, which does not change in response to the transgene. So this is the endogenous mouse ApoC-III in a transgenic mouse. And the gray, sorry, I got a text and I wanted to make sure you guys hadn't lost me. And then in the gray is the human ApoC-III. And so you can see the transgenic mouse has approximately 5-fold the ApoC-III expression in liver. And then this increase persists along the small intestine from the proximal to distal end. We initially did a lot of studies to check and make sure that there weren't changes to endogenous mouse ApoC-III gene expression either, and there aren't. There's just extra expression of human ApoC-III. If we look at plasma triglycerides, this is mgs per deciliter of plasma triglycerides in wild type versus the ApoC-III transgenic mouse, we see what we would expect. We see hyperlipidemia in response to high levels of ApoC-III. I want to point out that the average fasting triglyceride in our ApoC-III transgenic mice is between 1,500 and 2,000 mgs per deciliter. So often if you bleed these animals in the postprandial state when it's even higher, they have an almost Pepto-Bismol-like blood. If you compare this to wild type that are under 100, you can see the stark impact of high levels of ApoC-III on triglyceride clearance. So you're keeping a lot of those triglyceride-rich lipoproteins in the blood rather than having those lipoproteins be taken up by tissues and by the liver. We also looked at cholesterol. There's also a significant increase in plasma cholesterol. The difference is not as stark. You can see that it's about a 2-fold change. And the reason there is that ApoC-III really impacts the uptake of triglyceride-rich lipoproteins. Those triglyceride-rich lipoproteins do contain cholesterol, which is likely why you see this change, but they contain vastly more triglyceride. And so again, this is what we would expect from our transgenic model. We looked first at the immune milieu of our ApoC-III transgenic mice. We initially thought that we might see some hyperinflammatory markers. So in the left panel is the picograms per mil of circulating cytokines. I'm showing you a basic panel here of inflammatory cytokines like TNF-alpha, IL-6, IL-4, and interferon gamma. To be very honest, there wasn't anything particularly special about these 4 cytokines. When we started these experiments, we really just were brand new to immunology and wanted to look at some of the cytokines that we knew were inflammatory, and these were part of one of those inflammatory panels. We started and we saw no change between the ApoC-III transgenic and wild type mice in any of these cytokines. We've since assayed other cytokines and we find no differences. But when we looked at circulating IL-10, shown here on the right side between wild type and transgenic mice, and again, this is circulating, so this is plasma IL-10, we see a huge increase in plasma IL-10 in the ApoC-III transgenic mice, almost a 100-fold increase. IL-10 is an anti-inflammatory cytokine, so this was the exact opposite of what we expected to see, but was also seemed to be a result that was important based on the magnitude of this change. And so we have been trying to understand why we got so much IL-10 ever since we got this first piece of data. So we know IL-10 can come from a variety of cells. It can come from macrophages and dendritic cells. It can also come from regulatory T cells, which I'll call Tregs for the rest of this talk. I'm going to explicitly talk about CD4 positive, Foxp3 positive Tregs. These are the canonical CD4 lineage regulatory T cells. These are tolerogenic because they inhibit inflammation. Part of the way that they inhibit inflammation is by suppressing the proliferation and activity of effector T cells like Th1, Th2, and Th17 cells. And one of the ways that they do that is through their secretion of IL-10. They are not the only cells that secrete IL-10, and in some cases, they aren't even the major cell that secretes IL-10. But we went looking for cells that might have this property in our animals. And so I'll start here by showing you, this is the absolute number of splenic Tregs in wild type and ApoC-III transgenic mice by flow cytometry. You can see that there's an increase in the absolute number of splenic Tregs in our ApoC-III transgenic mice. And if we look at the canonical homing marker for the intestine, CCR9, we see that there's an increase in the number of Tregs that express that homing marker in the spleen of ApoC-III transgenic mice, which is a further hint that something's going on with Tregs and the Tregs that home to the gut. When we look in the intestinal lamina propria, that's this abbreviation here, in the lamina propria where the mesenteric lymphatics meet the intestinal epithelial cells, we see that there's also an increase in those Foxp3 positive Tregs in the ApoC-III transgenic mice. These are mice that are just on chow diet. They haven't been manipulated in any way. When we look at dendritic cells, just as an example here, and I haven't included all of the other cells that we looked at, I haven't included that data here, but when we look at dendritic cells, we see that there's no difference in the lamina propria in the total number of those dendritic cells. And again, these are the CD103 positive DCs that we would normally, that are normally found in the gut. We also looked at the total population of effector T cells. We looked at the total population of monocytes and macrophages, as well as dendritic cells and NK cells in the gut and elsewhere in the ApoC-III transgenic mice. And we find the only cell type that's different are the Tregs. There's no change in the total number of any of the other cells. And so we wanted to know whether these Foxp3 Tregs might express some, might have some differences in their expression of some of the immune checkpoint markers, trying to get at whether there was just a difference in accumulation or a difference in their actual character. And so we isolated splenic Tregs from our transgenic and wild type mice. We cultured them with and without T cell receptor stimulation, so that's anti-CD3, anti-CD28. And then we looked at their expression of these cell markers. And what you can see on the right is essentially the area under the curve. You can see that there's no difference both with and without TCR stimulation in the expression of these markers. And so we concluded from this that though we see an accumulation of these Tregs, the Tregs that accumulate are not different between the two genotypes, between wild type and transgenic mice. We also wondered whether the cells might proliferate at a different rate. And so again, we isolated those Tregs from the spleen of wild type and transgenic mice. We stained them with Ki-67 and then did a proliferation assay with and without TCR stimulation. And you can see here that there's not a huge change in the rate of proliferation. There is more, there's not more proliferation in either physiological context in the C3 transgenic Tregs. There are just more Tregs. And so again, in an early study, which we're showing here with the proliferation rate, we don't see a change in proliferation. So these data suggest that the ApoC-III transgenic mice accumulate intestinal Tregs. but they do so without a change in the Treg activation state. So we see that there are more Tregs, there's more plasma IL-10, but there really aren't, there's no other change to the Tregs. And so why are the Tregs there and what are they doing? And again, this has really been finding out why they're there and what they're doing has really been due to the blood, sweat, and tears of my previous PhD student, Kayla Rodia. And I'm going to continue telling you about her story there. So we asked whether these Tregs actually express lipid transporters and whether there was any enrichment in lipid transporter expression in the gut Tregs, since they didn't have any other major changes that we could measure. And so here I start with just Treg mRNA expression. So we isolated splenic Tregs and FACS sorted them and then looked for CD36, LDLr, and LPL. So some major canonical lipid transporter, and they do express the RNA for these. We also used an antibody against LDLr by flow cytometry to assess actual cellular LDLr expression, not just the mRNA, since we know that a lot of cells express mRNA for a variety of targets, but don't actually make protein. And we assessed how many cells, when we isolated them from spleen, skin draining lymph nodes, mesenteric lymph nodes, or the intestinal lamina propria, actually expressed LDLr. And what I hope you can appreciate from this graph is that in the intestinal lamina propria, so the closest that you can get to the intestinal enterocytes, you actually have a significant increase in LDLr expression in the C3 transgenic mice, and you also have the largest number or proportion of cells that are LDLr positive. And so if we look at it in a pie chart, you can see that in the spleen, we get approximately 72% of the Tregs express LDLr. As you move closer to the gut, to the mesenteric lymph node, 87%, and as you get to the lamina propria, almost all of them express LDLr. So we've been really intrigued with understanding what it is about getting closer to the intestine causes the Tregs to either increase their expression of LDLr, or what it is about LDLr expression that causes those Tregs to go to that anatomical location. We're definitely really interested in that. So I've shown you RNA and protein expression. I've shown you anatomical data suggesting that LDLr Tregs accumulate in the gut. Now we wanted to look at whether that LDLr was actually active. And so for this experiment, we needed a lot of cells. We isolated naive CD4 T cells from mice. We then analyzed BODIPI positive lipid uptake. And so I want to explain this experiment. So what we did was we gavaged wild type mice, donor mice, with olive oil containing BODIPI labeled triglyceride. And when you do this, 2 hours later, you can isolate blood and spin up the chylomicrons. And the chylomicrons that you isolate will contain some of that olive oil triglyceride, and they will also contain the BODIPI labeled triglyceride. So, so that was our experimental design. Adding on to this, we can also add recombinant ApoC-III, and I'm showing that here in red. And when we do that, we can make a pool of chylomicrons that are either derived from wild type mice and therefore have a medium normal level of ApoC-III expression, but also have the BODIPI labeled triglyceride. Or if we gavage ApoC-III knockout mice, as we did here, those chylomicrons can contain no ApoC-III. And so we've really got 3 populations of chylomicrons that we're assaying here. Again, we're assaying chylomicrons that are BODIPI positive but have no ApoC-III here. Another population of chylomicrons to which we've added recombinant ApoC-III here, and then wild type chylomicrons. What we would expect is that if you add chylomicrons without ApoC-III to a cell that contains lipid transporters like LDLr, that the lack of ApoC-III will cause that lipid to rapidly be endocytosed into the cell. And that's exactly what we see here. We find the BODIPI in the cell after the chylomicrons are added. When we add ApoC-III, we see a reduction in BODIPI triglyceride uptake. And again, that's due to ApoC-III's inhibition of lipid uptake. The wild type chylomicrons are intermediary because they do contain ApoC-III, but not as much as the chylomicrons here, to which we've added the same amount of ApoC-III as in our ApoC-III transgenic mice. So this experiment is really the proof of principle that the CD4 lineage expresses lipid transporters, that those lipid transporters actually transport lipid, they endocytose BODIPI labeled triglyceride, and that ApoC-III inhibits that event. And so that jives with our preliminary data in Tregs. When we do an in vivo experiment tracking BODIPI labeled lipid, in the mesenteric lymph nodes, we find something similar. So in this case, we've gated on BODIPI positive, Foxp3 positive Tregs. So we've isolated Tregs from the mesenteric lymph node after we've given a BODIPI gavage to these mice. And so I want you to focus here on the left hand. We have wild type mice. There is a amount of Tregs that are BODIPI positive. They're taking up that dietary lipid that's BODIPI labeled. And the ApoC-III transgenic mice have Tregs, have less Tregs with BODIPI. If we then wait, so this is approximately 2 hours after the gavage. If we wait 12 hours after the gavage, we find that still some of those Tregs contain BODIPI, but overall there's significantly less. So again, this is the in vivo proof of principle that Tregs can take up BODIPI triglyceride that has been delivered via an oral gavage. And again, this inhibition of BODIPI triglyceride endocytosis is consistent with ApoC-III's canonical role of inhibiting LDLr in all other cells. And so I think this is really important to then ask, what is triglyceride uptake doing in the Treg lineage? So we looked at Treg mRNA expression, and we looked at a variety of genes involved in lipolysis and beta oxidation, and we found that the ApoC-III transgenic Tregs had an increase in many of these genes. And again, this is RNA, so it just gives us a hint that there might be some changes to mRNA. To metabolic genes. We used seahorse metabolic flux analysis shown here. Here's the oxygen consumption rate over time. We used the canonical inhibitors shown here in red and blue, and we compared in the open circles wild type Tregs to, in the gray circles, the ApoC, or gray squares, the ApoC-III transgenic Tregs. And what we found is that at the early part of this flux analysis, we see that there's an increase in oxygen consumption rate. And if you move to the right side, the basal respiration is shown here, is essentially showing you that there's an increase in the basal respiration rate in the ApoC-III transgenic Tregs. And so that tells us that the essentially basal, or actually let me put that a different way, that these Tregs from ApoC-III transgenic mice that are potentially deprived of triglycerides are having to increase their basal respiration rate to make the same amount of ATP. And so we asked whether this change in respiration and triglyceride transport actually has a physiological role. And now I'm taking a break because a friend of mine suggested that every talk should have like a tiny break so that we can all take a deep breath. So I'm going to show you 3 pictures of my new campus. This is the University of Pittsburgh. This is the Cathedral of Learning. I'm not using this as my background fill, but I want to point out that this was built during the Depression, and the purpose of this building was to be a beacon of education during a dark time. And so part of the reason was to stimulate the economy during the Depression, and part was to bring communities together. And I think that having a cathedral dedicated to education is especially exciting for those of us academics. This is the Pittsburgh approved photo of the inside of the cathedral. I was flirting with the idea of using this as my Zoom background, but it didn't feel as personal as using my home office. Plus, this way, potentially you might get a cat or a kid infringing on my talk. Here's my own picture back in previous COVID-19 times when we all could get together and actually study. I love writing in this building. It's really gorgeous. Okay, so that's the end of the break. Now I'm going to actually show you guys some physiology that I'm really excited about. So I've told you that there are more Tregs in our ApoC-III transgenic mice, that those Tregs are high in the gut and especially in the lamina propria and in the mesenteric lymph nodes. I've told you that those Tregs express lipid transporters, that they take up differing amounts of triglyceride, that ApoC-III regulates the amount of triglyceride that they take up, and that by seahorse analysis, they have a functional change in metabolism. But who cares if they don't actually do anything? So we used the DSS colitis model to ask whether the accumulation of those Tregs was actually tolerogenic and could protect these mice from the strong effector responses that you see in response to DSS. So what you see here is that wild type and control mice were given 4% DSS water for 5 days. And in wild type mice, in the 5 days after that initial treatment, they will rapidly lose up to 20% of their body weight, mostly through diarrhea and through and because they're so sick that they don't eat. The ApoC-III transgenic mice, however, not only lose less weight, just over 10%, but they regain their original weight much faster than their wild type counterparts. And this is really consistent with an increase in the tolerogenic intestinal environment in the ApoC-III mice that occurs in result to the Tregs. When we look at the macroscopic damage score here, this is a qualitative assessment of how sick the wild type and transgenic mice are. And it takes into account how listless the animals are, shivering, and how much diarrhea they're having. And again, we take incredible care of these mice. These analyses are tough to do because DSS colitis really makes the animals sick. Our goal is always to reduce pain and distress and to use humane endpoints, while also keeping in mind that there are many, many Crohn's and colitis patients who are living with these symptoms today. Unabated. There are a lot of pediatric patients who die from these same events. And so we're using this as a model to try to come up with rational approaches to treating those people. So that's what the macroscopic damage score is. You see that the ApoC-III transgenic mice are significantly protected from DSS colitis and from its symptoms. When we look at markers of colonic tight junction maintenance, you see occludin and zonulin 1. Again, these are just two mRNAs in the colon that tell us a little bit about whether tight junctions are perturbed. We see that ApoC-III maintains tight junction expression, which is a sign of less inflammation in the colon. Also, colon length is maintained in the ApoC-III transgenic mice compared to the wild type mice who are quite ill. If we look at circulating IL-10, we see that there's still the maintenance of high levels of plasma IL-10 in the ApoC-III transgenic mice. This is likely the reason that these mice are less inflamed than their wild type counterparts. When we look for Tregs, we see that there's an accumulation of Tregs in the mesenteric lymph nodes of ApoC-III transgenic mice in response to DSS. And then when we look at LDLr knockout mice to try to get at whether this is an ApoC-III specific event or whether this is due to ApoC-III's inhibition of LDLr, this is when we see something that we think is really interesting, which is the wild type mice, again, after DSS, lose body weight due to diarrhea, whereas the LDLr knockouts lose significantly less. So if we compare the effect of total loss of LDLr to the effect of high levels of ApoC-III, which inhibit LDLr, we see that there's a real similarity in the ability to protect against DSS colitis. If we look at the macroscopic damage again in the LDLr knockout mice, they are significantly less ill. And if we look at the number of mesenteric lymph nodes, we see, or mesenteric lymph node Tregs, we see that they are increased. And actually in the LDLr knockouts, they also have an increase in total CD4 positive T cells, the parent T cells, which we don't know why this is. We don't know whether these are differentiating or proliferating more rapidly leading to this accumulation or not. This is part of the mechanism that we really don't know. We do know that we still see this accumulation of mesenteric lymphatic Tregs. And so again, these data are consistent with triglyceride transport through LDLr and inhibited by ApoC-III being a key factor in Treg accumulation and DSS colitis protection. Finally, we also used ApoC-III on chylomicrons as a therapeutic approach in mice. And so in this experiment, what we're looking at is body weight change. So the same way I've been showing you with the other DSS models, the same amount of DSS and then the same time period post-DSS to look at weight loss. And if you look in the black square, these are animals who received IP PBS and also DSS. They lose weight during the DSS colitis period like we would expect. When we give IP ApoC-III, so just protein ApoC-III, we see that there's also a significant weight loss comparatively with the PBS group. But if we give ApoC-III on triglyceride-rich lipoproteins, so the way that ApoC-III is found physiologically, again, we see that there's less weight loss during the DSS colitis induction period. And so ApoC-III on chylomicrons is able to protect these mice from developing the same level of colitis symptoms as the controls, the PBS, the mice that receive PBS, or ApoC-III alone. And so this really suggests to us that it's ApoC-III on a triglyceride-rich lipoprotein that is causing less inflammation in the gut. When we look at the macroscopic damage score, that is confirmed in the mice that received the ApoC-III containing TRLs. When we look at colonic gene expression here for Foxp3 and for IL-10, we see that it's also increased. We don't yet have the flow cytometry data looking at the total number of Tregs. We would expect that this might correspond to an increase in Treg accumulation. So in conclusion, what we see is that from our data set is that CD4 positive cells are able to endocytose lipids predominantly triglyceride from plasma triglyceride-rich lipoproteins, that these are the physiologically distinct and physiologically important triglyceride delivery vehicle. They are distinct from the less abundant pool of circulating free fatty acids, which are modeled here. We conclude that excess ApoC-III and the loss of LDLr stimulate IL-10 and intestinal Treg accumulation. And I want to asterisk here right now, the mechanism for this is unknown, and in large part the reason why I'm showing you this as unpublished data. Obviously there's a lot of interest in how the loss of LDLr causes Tregs to accumulate in the gut. We have some hints, but we're not exactly sure the reason. This set of data really led us to ask whether there might be consequences to the pharmacological lowering of plasma ApoC-III. So as I told you in the introduction, ApoC-III is a cardiovascular risk factor. ApoC-III stimulates plasma triglycerides. And for both of those reasons, there's a lot of interest in lowering ApoC-III so that you can lower people's triglyceride and cardiovascular risk. And we know that there are both antisense and monoclonal antibody therapies in the pipeline. This is from last year, showing that Novo Nordisk has bought an anti-ApoC-III antibody for its cardiovascular R&D group. So again, just evidence of the fact that ApoC-III is, and lowering ApoC-III is a pharmaceutical target. We used our ApoC-III knockout mice to just ask the simple question, are they worse off in response to DSS? So our transgenic mice are better off. The loss of LDLr makes mice better off to DSS, but what about the loss of ApoC-III? And so these are wild type mice compared to ApoC-III knockout mice. You see that there's no major change in total weight loss. When we look at macroscopic damage, however, we see that the ApoC-III knockout mice are slightly more sick. Again, this has to do with how much blood is in the stool, the amount of diarrhea, the amount they eat, how listless they are. So again, by qualitative measure, the ApoC-III knockouts are slightly sicker. When we look at the lamina propria Tregs in the colon, we don't see a significant difference, but we see that in general, the ApoC-III knockout mice really have very few colonic Tregs. And so we're actively looking into trying to understand how both the gain and loss of ApoC-III impact Treg accumulation. And so with that, I'm going to close. The takeaways here are that triglyceride uptake in immune cells is likely very important. It's the physiological delivery mechanism. We've shown in mouse models that this impacts physiology and in particular inflammatory bowel disease. We don't know the mechanism for those Tregs accumulating, but we do know that it involves LDLr and that there are changes to those Tregs metabolism. And since this audience certainly understands how intracellular metabolism drives cell function and whole body physiology, we definitely know that that's going to continue to be important when understanding the immune system. So I want to thank my funders, NIH, Cystic Fibrosis Foundation, and the Rainin Foundation for Inflammatory Bowel Disease. I always get questions about the CF Foundation funding. CF patients also have pretty significant intestinal defects in dietary fat absorption. And so I think I'm pointing this out just to say, if you're not interested in the intestine, I think it's a really, really interesting metabolic organ that plays a major role in whole body homeostasis. I want to thank the current members of my lab, And I especially want to thank the previous lab members who contributed significantly to this work. Diana Lee did a lot of the intestinal work showing ApoC-III metabolism in the gut. And Kayla Rodia did all the immunology work with all the DSS models, all the Tregs, and really drove this project. And so with that, I'll say this is a picture I took from one of the mounts in Pittsburgh. If you're looking for a postdoc, this could be you. You can find me on Twitter and at cohenlab.com, and I'll be really excited to answer any questions that you guys have. And I wish you all the best. Thanks.